Our next speaker is Meru Sadhu. He's from UCLA, from Lena Krugliak's lab.
Hello. So today I'm going to tell you about a new genetic mapping method that we've developed in the Krugliak lab. To begin with, I'd like to tell you about traditional mapping methods. So in genetic mapping, what you're generally interested in doing is if you have a trait that differs, say, in a population that differs genetically as well, you want to find the genetic variants that cause the difference in the trait. So what I'm depicting here is sort of a simplified diagram of linkage mapping, which is similar in philosophy to GWAS. So what we do here is, say you have your two parents that differ in some trait and differ genetically. If you cross the parents and then allow meiotic recombination to shuffle the genomes, well, you get our progeny that you can genotype to determine their shuffled genomes, and you can phenotype to determine their traits. And what you want to do then is look for regions of the genome that associate with the trait. So in this case, what we can see is that between these dashed lines, if you have the purple genotype, you have the trait. If you have the orange genotype, you don't have the trait. So what you've done is you've mapped the trait to this region of the genome. This is great, and it works very well. One challenge, though, is that these regions, their size is determined by the local recombination rate. And so if you now have this region that you've mapped and you want to say, great, let's map it some more, or let's get some more resolution, that can be challenging because you're dependent entirely on the recombinations that occur naturally. So what we wanted to do was see if we can sort of speed up this process by directing recombinations where they're most informative. And the way we're doing this is using CRISPR. Very quickly, the way CRISPR works is you have this endonuclease Cas9. It cuts DNA, and where it cuts DNA is dependent on a guide RNA that it binds. So if the targeting sequence of the guide RNA matches somewhere in the genome, Cas9 will cut there. And the reason we thought this could be useful for us is that recombinations actually start with a heterozygous DNA double-strand break. So typical recombination works through repair of that double-strand break off of the homologous chromosome, and that leads to a crossover where that double-strand break occurred. So if you can direct double-strand breaks heterozygously, you can get recombinations. And another nice feature of directing breaks in this way is, so what I've depicted here looks like meiosis, but it actually also works in mitosis. If you have a DNA double-strand break in mitosis that's repaired the same way, you can still get a crossover, and the outcome is a little bit different, where ultimately you get what's called the loss of heterozygosity, and the way that works is if one of the recombined chromosomes, say here, segregates the same daughter cell as a non-recombined chromosome, you get a genotype that has the same heterozygous genotype for most of the genome, but from the recombination point here, say, to the telomere, you become homozygous for one genotype or the other. Okay, so what we wanted to see is, A, can we even direct recombinations in this way, and then if we can we use this to map traits? So the system that we're using is yeast. We do our experiments in this cross between a lab yeast that we call BY and a wine yeast RM. We focused on the left arm of chromosome 7, and what we did is we targeted using guide RNAs with different targeting sites, Cas9 to cut sort of all across the left arm of chromosome 7. And we have some sites that cut specifically on the BY chromosome and some that cut specifically on RM. What we did is we selected about 4 colonies per targeted site for 384 total. And then to see where the recombinations occurred, we sequenced their genomes. So what I'm showing here is where we've determined the recombinations occurred on the y-axis, plotted against where we targeted the cuts to occur. And what you can see is that by and large, there's generally good concordance, where 75% of the recombinations occur within these dashed lines, which demarcate 20 kb around the targeted site. So that's great. We can target recombinations, which was the goal. Another thing you might notice is that the recombinations don't occur precisely on top of where we targeted. This is expected because in the repair process, what you get is holiday junction migration, and so the actual crossover moves a bit relative to where the double term break occurred. In our case, this is kind of nice because what this means is by targeting 100 unique sites, but collecting more than 100 unique individuals, we get more than 100 unique recombination points. Okay, so now that we've made the genetic variation, we want to see if we can see any phenotypic variation and then map that phenotypic variation. So the way we do our phenotyping is by taking our yeast and pinning them to various challenging conditions, letting them grow, and then seeing the size of the resultant colony and using that as a trait. So in this case, what I'm showing is growth on manganese, which is a metal stress. You can see that some of these LOH colonies can grow pretty well, and some of them grow pretty poorly. So there is phenotypic variation. Now, to map the phenotypic variation, I'm going to show the data with these plots, where on the y-axis I showed the colony size on manganese sulfate, in this case, and on the x-axis I'm showing the position of the breakpoint, the loss of heterozygosity. So in particular, points that fall closer to the left end, their breakpoints occur closer to the telomere, and if the points further to the right, their breakpoints occur closer to the centromere. And then the last important bit of information on these graphs is the color of the points. So if the point is purple, then the loss of heterozygosity gains RM in the homozygous region, and if the points are orange, they gain BY. Okay, so here's all the data. And so the way we interpret these graphs is, so we move in from the telomere. So what you can see is that all of these individuals that have breaks within, say, the first 190 kb in from the telomere, if you become homozygous BY or become homozygous RM, your phenotype is not affected. Your trait is the same. So what we can conclude is that there aren't variants between BY and RM in this region that actually affect manganese sensitivity. However, past about 190 kb, you can see that things changed quite drastically. So there's something that changes between BY and RM, that if you become homozygous RM for that variant, you become actually very resistant to manganese. And if you become homozygous BY for that variant, you become sensitive to manganese. So sort of we can visually map that to about 190 kb in from the telomere. We can also do this more quantitatively by calculating a LOD score and using that LOD score to calculate a confidence interval. In this case, what we map this variant to is about 3 kb on this chromosome arm. So we've used these targeted breaks to map manganese sensitivity to about 3 kb. That's great. In these 3 kb, however, there are still several variants between BY and RM. And what we wanted to do was ask, well, can we target more breaks just in that area to determine which of those variants really causes the trait? So we did precisely that. This time we used guide RNAs that rather than targeting across the entire chromosome, we targeted really just in that vicinity. And we picked this time 378 colonies with LOH events sort of targeted in this area. And again, genotype by sequencing. And in this case, so what I'm showing here is a plot of the recombination rate by physical distance in this vicinity. And overall, what we see is that in our targeted individuals, 13% had a break within the 3 kb interval. This is actually hotter or more recombinations than you'd see at any natural recombination hotspot in yeast. And in this particular interval, this actually turns out to be sort of a recombinational cold spot in yeast. That's what I'm plotting in red. So in normal meiotic recombinations, you see fewer than 1% of individuals would have a break in this area. Okay, so now that we have these really targeted genotypes, let's map their phenotypes. So I'm showing the same sort of plot as before. This is, however, with a second panel of individuals with targeted breaks. And also, this plot is really zoomed in. So before we were looking at about 1 or half a megabase. Here we're looking at just 10 kb around the interval that we mapped in the first round. And what we can see, first of all, is that there are lots of points within these dashed lines. So these are all points that correspond to individuals whose breaks occurred in the first interval. And at the bottom here, I'm showing the polymorphisms between BY and RM. And what you can see is that this polymorphism right here, if that polymorphism is heterozygous, as is the case for all of these guys, right, who have their loss of heterozygosity closer to the telomere, If that polymorphism is heterozygous, the individuals have the intermediate phenotype. But if this polymorphism becomes homozygous, either for BY or M, then you have the sensitive or the resistant phenotype. So again, visually it looks like we can map the trait now just to this one polymorphism. Once again, we can calculate a LOD score with the gray line and calculate a confidence interval. And indeed, the confidence interval for this trait now is just the one polymorphism that seems to really cause the trait. What we have done is gone and really confirmed this directly. We made this mutation, introduced it from RM into BY, and saw that it conferred resistance to manganese. The polymorphism happens to be in this gene that's a manganese transporter, PMR1, and it mutates a conserved leucine to phenylalanine, which is a pretty disruptive mutation. Okay, so what I've shown you so far is that we can use our method for rapid fine mapping of traits. We're also excited about another application of this method, which takes advantage of the fact that these breaks that we're generating don't require meiosis, as is the case for traditional mapping. All of our breaks that we've been using so far occur in mitosis. So where we're hoping to apply that is in mapping across species. So generally speaking, we have a poor understanding of the genetic basis of differences between species, phenotypic differences between species. And that's because traditional mapping methods rely on meiosis to generate the recombined chromosomes you need for doing mapping. However, there are many cases of species where you can form interspecies hybrids. These hybrids are often sterile, so they don't give you the recombined chromosomes you need, but you can form the cells at least that have both pairs of chromosomes. So if in these cells that have both pairs of chromosomes, if you can get mitotic recombinations, then in theory you could use our method to map traits across species. So cerevisiae happens to be one of these species that has related species that I can form hybrids with. These hybrids are all sterile. It's actually a pretty large complex of species that can intermate. And what you get are diploid cells that have both sets of chromosomes. They can divide mitotically fine, but you can't pass them through meiosis successfully. So what we did is we mated cerevisiae paradoxus. They're sister species, but they're actually quite genetically diverged. They're about 10% diverged at the DNA level, which is a lot more than your typical pair of sister species in the animal kingdom, for instance. So what we did is we targeted, once again, 100 cut sites to the left arm of chromosome 7, and this time we picked just over 1,000 loss of heterozygosity colonies. And once again, we genotyped by sequencing, and what you can see is that our breaks, once again, are pretty well targeted. So the majority of individuals have breaks within 20 kb of the targeted sites. Okay, and then I only have time to walk you through one phenotype, but what we've been doing since this time is taking these individuals and pinning them once again to various conditions and seeing whether we can see any correlations between genotypes and phenotypes. So here's an example trait. This is lithium chloride. And what I guess I want you to take away is that there's a lot going on, even just in this one chromosome arm. So in this one chromosome arm, which is about half a megabase, what we see is 3 loci that control differences between cerevisiae and paradoxus for lithium sensitivity. And in particular, one thing that I'm excited about is that they don't all go in the same direction. So this first gene here, the cerevisiae allele gives you sensitivity, whereas these other two, the cerevisiae allele makes you more resistant. Okay, so quick summary, we're using CRISPR for genetic mapping. This allows rapid fine mapping because we get targeted recombinations, and we're using it now for interspecies genetics. And with that, I'd like to take any questions.
So we'll have to move on. We're out of time.